Today, we're diving into a story about a brilliant molecular shortcut and a tiny fluorescent snitch. It's a breakthrough that could completely change how we detect dangerous viruses, giving us lab-quality results super fast, anywhere in the world. So let's just jump right into the big problem this technology is trying to solve. I mean, think about it. Imagine you're a shrimp farmer and your livelihood is being wiped out by a mysterious disease, or you're a doctor in a remote village during an outbreak. You need answers, and you need them like yesterday. How on earth do you get a reliable diagnosis without a high-tech lab? And that brings us right to our first section. We're talking about a huge challenge in global health and agriculture called point-of-care diagnostics. The goal is pretty simple, right? Bring the test to the patient, or in this case, to the shrimp, instead of having to drag the shrimp all the way to the lab. And this slide really lays out the massive gap. On one side, you have your traditional lab test. Yeah, it's powerful, but it's also painfully slow. It needs all this expensive, bulky equipment, not to mention a team of experts to run it. But what you actually need out in the field is the total opposite. Something fast, cheap, easy to use, and small enough to carry with you. Okay, so to try and bridge this gap, scientists came up with a really clever shortcut. It's a technology called insulated isothermal PCR, or just iiPCR for short. The whole idea is to make one of the most powerful tools in biology way faster and way simpler. So first, a really quick refresher. PCR is pretty much the gold standard for finding a virus. You know, if a virus's genetic code is like one single page hidden inside a massive library, PCR is like a molecular photocopier. It makes millions and millions of copies of that one page until it's super easy to spot. So how in the world does ePCR speed this whole process up? Well, the secret is actually physics. Instead of some big, complicated machine that heats and cools the sample over and over, it uses this natural process. By just heating the bottom of a tiny tube, you create a natural current that automatically cycles the DNA through the hot and cool zones it needs to make copies. It's basically a tiny, high-tech lava lamp for DNA. Super cool, right? But here's the catch. The lava lamp made copying the DNA a lot faster, sure, but it didn't solve the whole puzzle. To actually see if you'd copied anything, you still had to take the sample and run this other slow, messy lab procedure to read the results. So you were still kind of chained to the lab. And this is where we get to the real breakthrough. To finally cut that last tie to the lab, the researchers added this absolutely ingenious little molecule. It acts like a fluorescent informant, a tiny molecular snitch. Now this quote from the paper, this is the secret sauce. What they did was use a special tool called a TaqMan probe. You can think of it like a tiny guided missile designed to find the virus's DNA. It's got two parts, a tiny light, called a reporter, and a little cap, called a quencher, that keeps the light turned off. The magic happens when that molecular photocopier we talked about runs it over. Okay, this slide breaks down this beautiful little four-step dance. First, the probe, our snitch, finds the virus's DNA and latches on. Then, as the DNA photocopier is doing its thing, chugging along the strand, it runs right into the probe. Step 3, the copier smashes the probe apart, which knocks the cap off the light. And finally, the uncapped light glows. A glowing sample means the virus is there. No glow, no virus. It's that simple. So you've got your fast photocopier and your glowing snitch. The next logical step was to put them together in one device and see if it could actually handle a real-world threat. And here it is. This is the diagram from the paper showing the whole device. On one side, you've got the iiPCR module, our little DNA lava lamp, and on the other, they've built in an optical system, which is basically a tiny camera with some LEDs and filters designed to do one thing: look for the glow from our snitch. The whole thing is packed into one neat, compact unit. For their test, they picked a really nasty target, white spot syndrome virus, or WSSV. This thing is a huge deal in aquaculture. It can literally wipe out an entire shrimp farm in just a few days. So yeah, this was the perfect high-stakes challenge for their new gadget. And the results? They were absolutely stunning. The test turned out to be incredibly sensitive. It could get a positive result from as few as 10, that's 10, copies of the viral DNA in a sample. To put that in perspective, that's like trying to find just 10 specific typos in the entire Encyclopedia Britannica. But look, being sensitive is only half the battle. The real question is it accurate? So to find out, the researchers set up a head-to-head showdown against the officially certified industry-standard test. They took 120 real-world shrimp samples and tested them with both methods. And honestly, the results on this chart speak for themselves. The new test was a home run. It had 100% sensitivity, which means it caught every single positive case. Not a single infected shrimp slipped through. And its specificity was almost 97%, which means it was extremely good at correctly identifying healthy shrimp. So basically, no false alarms. It was both incredibly sensitive and super reliable. So what's the big picture here? What does all this really mean? Well, this is about so much more than just protecting shrimp. This is a potential blueprint for the future of how we fight disease. Any disease, anywhere. This table from the study really puts it all in perspective. Just look at the top row. The new method gets you an answer in 35 to 40 minutes. Now, compare that to the old standard on the bottom, which could take up to 3 hours. That means it's about 4 times faster. And for a very similar price per test. That's a huge leap forward. So the ultimate takeaway is pretty clear. What we have here is a system that gives you the accuracy of a sophisticated laboratory, but it does it in a simple, portable box in less than an hour. No more complicated steps, no more waiting around. The answer is just a simple glow. And this really leaves us with a fascinating question to think about. This research proves that we can pack the power of a molecular diagnostics lab into a device that can fit in a briefcase. So the question isn't if we can do it anymore. The real question is, where do we take this power first? To remote clinics? To airports? To farms? The possibilities are just absolutely transformative.